Good afternoon, very good afternoon to all and one. We are really very privileged to have amongst us an eminent speaker, Dr. Supriya Ratnaparkhe. She has been done her masters with medal from Gujarat University and PhD in Biotechnology from Virginia Tech. Thereafter, did her postdoc at Georgia Tech, Atlanta and University of Georgia, Athens. Her postdoc research experiences have been in diverse fields, from tissue culture to cancer biology, to biofuels and bioenergy. Her research work is published in several reputed journals including Planta. Thereafter, did her postdoc at Georgia Tech, Atlanta and University of Georgia, Athens. Her postdoc research experiences have been in very diverse fields, from tissue culture to cancer biology to biofuels and bioenergy. Her research work is published in several reputed journals including Planta, PNAS, having an impact factor 6 and molecular cancer research besides several others. In 2012, she joined Institute of Chemical Technology, Mumbai as DBT Energy Biosciences Overseas Fellow. She was also involved in several national and international projects of BARC, including the development of India's first 2G ethanol technology in collaboration with BARC. Later, she pursued her DBT Energy Bioresources Overseas Fellow at Biotechnology, DAVV, Indore. Presently, she is Head Research and Development, Indore, Biotech Inputs and Research. Her research revolves around biofertilizers and biopesticides and other organic inputs. Dr. Ratnaparkhe has attended several seminars and conferences and been as invited speaker in various international and national conferences. She has also been a reviewer for several scientific journals on editorial board of some. She has also been the recipient of grant for 3 national and international projects in capacity of principal investigator or co-principal investigator. To add more feathers in her cap, she has been the recipient of the most prestigious DBT Energy Biosciences Fellowship at DBT ICT Center for Energy Biosciences. Her Best Poster Award at Jordan Research Conference, Boston, MA, USA is also amongst your accolades. We are privileged to have such an eminent speaker amongst us. We welcome you to enlighten us with your knowledge. Supriya ma'am.
Thank you so much, Bhavita, for such a wonderful introduction. It was really nice. And I congratulate the organizers for putting together such a good event. And thanks to Dr. Sai Prasad and Dr. Sharma for a very good and enlightening, very informative lecture. So now I'm going to present today's lecture from industrial perspective. I would like to share my screen now. Am I audible? Am I audible?
Yes, ma'am, you're audible. Yes, ma'am, you're audible.
And now, let me share my screen. Can you see the screen now? Can you see
Yes,
it now?
yes, ma'am, it's visible.
Yes, good. Full screen is visible now?
Yes, ma'am.
All right. So I'm going to speak about biofertilizers. And in fact, I would like to include some other microbial inputs as well today. From lab to commercialization, from an industrial point of view, because we have already heard such good lectures from Dr. Sai Prasad and Dr. Sharma, and they have elaborated on policy matters and a lot of commercial aspects as well. So a very practical industrial point of view will be presented in this lecture. And hello to all, greetings to all. I'm Dr. Supriya Ratnaparkhe, and I'm Head Research and Development at Indore Biotech. Our company has been serving the industry of biofertilizers for more than 26 years now. Just a small overview of our company. We cater to the needs of farmers in almost 60% of India, and we have our state branches in 7 different states of India, and our certificates and credentials, we have achieved these through our products and our performance in the market. We have achieved these certificates. And our latest is a recognition to our R&D activities from DSIR. Now, I have been asked this question by many people, and many people feel that organic manure, compost, all this is also biofertilizers and biofertilizer. And I do agree. Even I used to feel that compost, manure, this is also biofertilizer because it is biologically derived fertilizer. It's such a wonderful fertilizer. However, after joining here at Indore Biotech, I realized that there is a difference. The government has put demarcation between biofertilizer. It has very clear definition for biofertilizer, biopesticide, organic fertilizer. These are all organic inputs, but they have been put under separate categories. So biofertilizers, when we talk about biofertilizers, as the earlier speakers also said, that these are live microorganisms that are used to inoculate the soil, and they are helping in nitrogen fixation, phosphorus solubilization, and nutrient solubilization, as well as other mineral solubilization, as well as mobilization. Now, NPK, they come under the purview of macronutrients, and there are other micronutrients also. So biofertilizers' job is to provide all these nutrients, and these are live microorganisms. Organic fertilizers, like when you talk about compost or manure, that organic fertilizer is made up of one or more unprocessed material and may include unprocessed mineral material also. And that has been altered through microbiological decomposition process. So here I would like to point out an earlier question where one of the participants asked what integrated farming, how integrated farming can be helped with the help of biofertilizers. Now, there are some products in the market which are sold by the name PROM, phosphate-rich organic manure, or biopotash. Now, these products, especially PROM. So PROM, it is a technology rather than a product. So in this technology, what they have done is that they have used phosphate solubilizing microorganisms to decompose phosphorus, inorganic phosphorus compounds. So what they do is they enrich the waste with inorganic phosphorus compounds, and they add phosphate solubilizing bacteria or phosphate solubilizing microbes, any microbes, to make that particular compost into a phosphate rich organic manure. And similarly, you can do with potassium rich potassium solubilizing bacteria as well. So there are biofertilizers, there are organic fertilizers, there are biopesticides. Now biopesticides also, you can have live microorganisms such as trichoderma, there are pseudomonas, and I will be touching upon this category also later on in the lecture, because this is also very interesting. Biopesticides, there are certain microbes which are multi-trait as well. Okay, so they act as biopesticides as well as biofertilizers, and that is why I asked the question earlier, is it mandatory to mention on the biofertilizer label whether is it mandatory to mention the microbe that is being used as biofertilizer, because many biofertilizers also act as biopesticides. And if we mention the name, the strain, the species, or the genus, it will come in the biopesticide category as well, such as Trichoderma viridis, Trichoderma hasianum. These are very good biofertilizers as well as biopesticides. Pseudomonas fluorescens, although well, yeah, there are certain Pseudomonas species which are very good mineral solubilizers also. Then there are biostimulants. Biostimulants are microorganisms are not currently, they are not included in the purview of biostimulants, because but there are several microorganisms that act as very good biostimulants but do not act as biofertilizers. So the companies are kind of confused right now where exactly to place them, whether to sell them as biostimulants or as biofertilizers. So these are just, formal categorization of agricultural microbes that have been done by government for ease of marketing as well. Probably the policies were made for easing the marketing of these products. Anyways, so biofertilizers, how are biofertilizers useful? The speakers have already explained in detail how biofertilizers help the soil as well as plants. We have noticed in our observation is that the soil that has been fed with inorganic fertilizers over a period of time, it is poor in quality, in the sense it has lower cation exchange capacity, it has lower water retention capacity, the microbial diversity, because you're smothering the soil with addition of so much of chemicals, that the microbial diversity is definitely going to be affected negatively. Then you have to increase the amount of herbicide to be added to that soil. You have to increase the pesticides and fertilizer input. Now, whatever fertilizer, like the inorganic fertilizers that are mostly used are urea. There's also DAP. For phosphates, for nitrogen, you add urea. And most of that fertilizer which is added to soil is not used up. And then what happens is it starts accumulating, it starts converting into fixed forms, starts making compounds that are not usable, that are not available to the plants. And so what you have to do is go on adding more and more to it. So eventually, at the end of it, if you look at it, you have spent more on inorganic fertilizers than you would have expected, just to get that kind of yield, because they are giving you quick response. You think it is very good, but you are not understanding that you have to use a lot of it over a period of time, and you are depleting the quality of your soil in the process. What these biofertilizers do, you will not see the response early on, but what you can do is you can combine these with compatible inorganic fertilizers. I'm saying compatible because the inorganic fertilizers should not kill your microbial population. The biofertilizers that you are using as bioinoculants should not be affected, right? You want them to survive, thrive, and flourish in the soil. That is the reason you are adding biofertilizers. Now, these biofertilizers should be able to convert the unused inorganic compounds also into usable form in your soil. And that is what will happen. And while doing so, they will grow in population and they will improve the quality of your soil in the terms of cation exchange capacity, water retention, the texture of the soil that has been fed with organic fertilizers is way better than that which has been fed with only inorganic fertilizers. The Rangwasa Jaivik farm that we have near our manufacturing unit can boast of not having been treated with any inorganic, any synthetic fertilizer or pesticide in more than 26 years. That is what we have done. That is a very big experiment that our managing director has done on that farm, that it was never treated with any synthetic chemical. No chemical input was put in it, and we got the soil of our farm analyzed, because we were going to have a visit from Honorable Sumitra Mahajan, Mrs. Sumitra Mahajan. So at that time, we got our soil tested, and this soil turned out to be such fertile, such high in carbon content. The carbon content is very, very important for any soil. We had very rich carbon content, and all the nutrients were in abundant amount in the soil. We have noticed that we do not have to use herbicides, very few pesticides, and that too biopesticides can suffice. Fertilizer input, we don't have to recharge the soil again and again with the use of fertilizers. So that is what the biofertilizers are doing to your soil. It renders plants resistant to biotic and abiotic stresses. It improves the soil fertility, texture, and nutrient content over a period of time. I cannot stress enough on the term, on this over a period of time, this phrase, over a period of time. So you'll have to wait. You'll have to be patient when you start using biofertilizers. The market for biofertilizers is definitely increasing, and it has been projected by 2027, it will have raised by 14.27%, and it is assuming that the pandemic is over, because pandemic has taken toll on many industries, including any fertilizer industry. And the global trend is that bacterial inputs, amongst all the microbial inputs, bacterial inputs market is going to be highest. And amongst formulations, as the earlier speakers told you, that there are carrier-based solid formulations as well as liquid formulations. So amongst formulations, liquids are more preferable because of the ease of use and many other advantages that they provide. Then, of course, the government is also taking a lot of interest these days on natural farming. In India, natural farming is promoted as Bharatiya Prakritik Krishi Paddhati program under centrally sponsored scheme Paramparagat Krishi Vikas Yojana. So if anybody is looking to come up with a startup in a related field, now is the time. It is a golden time for organic fertilizers industries, or any industry that is related to organic farming. It is the time is ripe for them. And there are many different fields that are related to organic farming where you can start your where you can think of a startup business. And government is also giving a lot of subsidies and a lot of aids. The banks are also giving you very good support, and you can approach NABARD, which is also a body which funds this kind of projects. And you just have to come up with a good project and you can approach them. Not only that, if you feel that you have some very good product that you can put in market, you can approach BIRAC. DBT's it is a DBT's branch which helps industrial projects. And it is roughly estimated that around 2.5 million farmers in India are already practicing regenerative agriculture. Regenerative agriculture is natural agriculture. In the next 5 years, it is expected to reach 20 lakh hectares in any form of organic farming, including natural farming, of which 12 lakh hectares are under Bharatiya Prakritik Krishi Paddhati program. So the government is also this natural farming has become one of the major focus of government mandates. So you have already been familiarized with the types of biofertilizers. There are nitrogen fixing microbes. Now these microbes, nitrogen fixing microbes, can be free living, they can be associative, they can be symbiotic. Among symbiotic, rhizobium and of course Frankia, and these are very commonly used. Rhizobium is the most used amongst symbiotic nitrogen fixing microbes. Associative, in associative, azospirillum, which is very good, it associates with rice rhizosphere. And free living are azotobacter and nostoc, Klebsiella nostoc is a cyanobacterium. And there are some very good other cyanobacteria also, such as Anabina and many others that are helpful in nitrogen fixing. And then there are phosphate solubilizing or mobilizing bacteria. Most of the industries like to use bacilli for this purpose, because bacillus produces spores. Bacilli are spore producing bacteria, and spores are easy to store. Spores have a longer shelf life, so most industries like to use bacilli for this purpose. There are zinc solubilizing microbes. Solubilizing microbes. Mycorrhiza is a wonderful microbe. It is a fungus that is endophyte. And mycorrhiza, VAM, especially vesicular avascular mycorrhiza, that is very much in demand in the market right now. And there are potassium solubilizing or mobilizing bacteria, sulfur oxidizing, and plant growth promoting rhizobacteria are not currently under the purview of FCO, but as Dr. Sharma said, they might soon be included there. So these are the biofertilizers, main biofertilizers that have been mentioned in FCO. So nitrogen fixing bacteria, nitrogen fixing microbes, I would rather say, essentially need to have nitrogenase enzyme, which they use for fixing the bacteria. So what they do is they convert atmospheric nitrogen, which is not usable by plant. It is not in the usable form by the plants. And so what happens is that is converted to ammonia with the help of nitrogenases. Okay, so that is what they do. And you all must be very familiar with how these diazotropes act, because you have been studying microbiology and biotechnology. So this is a very common phenomenon. Now, mineral solubilizing or mineral mobilizing bacteria or microbes. what do? There's a very wonderful micro ecosystem at work in the soil. What this micro ecosystem means is that the plant roots that are inside the soil, they they secrete sugar, amino acids, and vitamins, which are taken up by these microbes, which in turn are producing certain enzymes such as phosphatases, um, or they are producing organic acids. These organic acids or enzymes or inorganic acids, which are being produced by these bacteria or other microbes that are present in the soil, these help in breaking up the soil minerals as well as the complexes that have been made by inorganic fertilizers inside the soil. So those are broken down into available form, which are then taken to the root by mineral mobilizing bacteria. And then that helps in uptake of these nutrients by the plants. So this is how this micro ecosystem works. Conversion of unavailable nutrients into available nutrients, and then mobilization of it and uptake of it. So that is how this micro ecosystem works, and it is enriched by addition of biofertilizers. Vesicular avascular mycorrhiza, this I told you earlier, it is a wonderful biofertilizer. As you can see, with the use of mycorrhizal fungi, you can see the kind of root system plants are able to have, and without mycorrhizal fungi. And this picture over here, sorry, I'm not able to use my pointer here, but this picture over here of the root, the comparison between two types of roots, this is with our own product, where the root system has developed so much better compared to where mycorrhizal biofertilizer was not used up. So what mycorrhiza does is, the spores of mycorrhiza that are present in the soil, they send their mycelia inside the root hair. Okay, root hair, roots, they send these mycelia inside through a this thing, infection point. There is an infection point through which the mycelia enter the roots, and this way there is a bridge between the soil and the root itself. Okay, between the plant and the soil. So that makes it easier for the plant to uptake the nutrients from its rhizosphere. So this is what mycorrhiza is doing, and mycorrhiza also improves the root mass. Okay, that way the network of mycorrhiza mycelia, it improves the root mass as well. So that soil becomes richer in moisture resistance also, better in moisture resistance also. So it becomes resistant to drought stress, and many biotic stresses are also mitigated with the help of mycorrhiza. Mycorrhiza also helps in solubilizing phosphorus. So there is a very big list of benefits that mycorrhiza provides to the plants and to the soil. So the texture of soil that has mycorrhizal growth is much better than the one without it. In these two pictures at the bottom, you can see how the vesicles of mycorrhiza and arbuscules, the mycelia and the vesicles that enter the root, these are sections of roots in which mycorrhiza has already infected. Okay, so this is just, I told you that I'll be touching upon biopesticides with just a single or a couple of slides. So biopesticides, since Dr. Sharma has already talked about biopesticides in detail, this is just to give you an idea of the major biopesticides that are in market right now. This is a viral biopesticide, nuclear polyhydrosis. virus. This is highly effective against Helicoverpa amigera, which is a very bad pest. And what this does is that this virus, it kind of solubilizes the midgut, alkaline midgut of the insect that comes in contact with this virus. And a good thing about it is, when I say that biofertilizers and biopesticides are sustainable systems, a good thing about this is, and we have used it many times, that when the insect comes in contact with this virus, and the virus gets into the body of the insect, it multiplies within the body of the insect, kills the insect, but you can always take that insect, because these are viral cells. You can always take that insect and just crush it and use it as biopesticide itself, the body of the dead insect, the dead larvae, dead bodies of dead larvae of Helicoverpa amigera can be used as biopesticide, the crushed part of it. So this is a wonderful sustainable system here. Then there are certain fungal pesticides. So what these fungal pesticides do, they affect, they infect the insects that they come in contact with. The spores of these fungi, they come in contact with insects, and then they produce certain enzymes such as chitinases. You know that the body, the shell of insects are made of chitin. So these are very good at, this particular example is Bavaria bassiana, and it is a very good chitinase producer, which infects the insect and it kills the insect that it comes in contact with. So this is also very good pesticide which is in market. Then there is BT. Pretty much everybody knows about Bacillus thuringiensis and the cry protein that it produces, which binds to a receptor in the gut of microbe in the on the membrane of the larvae. And what it does is it enters the larvae. These cry proteins are released, and they destroy the gut of the larvae. So this is how BT works. And this is also another very good pesticide that is in the market. And these, you won't believe me, and I have showed you Trichoderma viride here, but Trichoderma viride is a very potent fungicide. And the pure spores of Trichoderma viride, the pure spores of Bavaria bassiana, these pure spores sell for such a high price, 4 to 40,000 rupees per kg in the market, depending upon the purity of the spores. So you have a very good market for these fungicides and pesticides. And so therefore, I feel that this is a very profitable business as well. Of course, this is antipedin. This is non-microbial pesticide. As I directed, these are just derivatives of live organisms. Now, if we come to commercial aspect of it now, in commercial aspect, now what you have to do is, in order to commercialize a biofertilizer or biopesticide product, you have to isolate that particular microbe. You have to identify, characterize. So these things are done in the lab, okay, the laboratory. Then there are commercial aspects of it, mass production, field trial, registration. These are done in the industrial environment. And there are certain things that can be done in both the environments, such as formulation, bioefficacy, field efficacy, quality parameters. So these two are like research part of it and commercial part of it. Now, this slide kind of summarizes how you carry out isolation, identification, characterization. So what you do is, if you isolate a certain microbe, and you want to commercialize that microbe, if you feel that, okay, this is showing some activities of biofertilizers, and you want to commercialize that microbe, then what you do is you isolate that microbe. First and foremost, you carry out certain tests such as biochemical tests. Then you have to optimize the growth parameters. You have to characterize it on selective media, like for example, for phosphate solubilizing bacteria, you use Picoskaya. For potassium solubilizing, you use Alexandrovs. There are several media that are available in the market for selection of these bacteria or funguses. And then what you do is, depending upon what you're wanting to commercialize. So there are bacteria, there are fungi, and what kind of activity you're looking at, whether you're looking at antagonistic activity, whether you are looking at the mineral solubilizing property of it. So accordingly, you will carry out the characterization, the biochemical tests, you will optimize the growth parameters, and then you will send it for identification. 16S RNA, 18S RNA is the most common method of identification. Sequencing method is the most common identification technique that is adopted at present. And then you carry out media optimization. Here I have shown liquid media. You can also do solid state fermentation. So you have to optimize the media in which your microbe is showing best growth. Then you have to also study the life cycle of that microbe. That is very, very important, to study the life cycle of microbe, because that will be helpful in scaling up the production of that microbe. Then you can carry out pilot scale, 20 liters and above. So you can carry out pilot scale culture of that microbe. That will give you an idea whether this media that you have optimized, or the technology of culture that you have optimized, whether it is scalable or not. Okay, because unless it is scalable, you cannot apply it in industrial environment. So it is very, very important to make sure that this technology is scalable. So these are the kind of things that you can do. Now, if you do not want to go through all this, you do not want to isolate your own microorganism, there are several institutes and national laboratories in India that have developed these technologies, and they can readily give them to you. And government even has subsidized technologies, subsidized prices for these technologies that they offer to MSMEs. And you know, they have various schemes for various industries, and they can transfer these technologies to you. For example, ICAR transfers all its technologies through this portal called Agri Innovate. So you can go on Agri Innovate, you can look at all the technologies that have been developed by various ICAR institutes, and if you're interested in any of them, you can express your interest, and then you can carry on the transfer formalities. Then there is BARC also. BARC also provides some very, very good agriculture related technologies. They have some very good trichoderma culture technologies, VAM, and many such technologies that they transfer to industries, and they also have subsidized rates for MSMEs. Then there are universities, agriculture universities, such as Tamil Nadu Agriculture University, Assam Agriculture University, who have developed very good technologies. So you can buy those technologies from them. And there is this National Center for Organic Farming, which provides very good training programs. The ICR Institutes also provides very good training programs for organic farming. And they have various modules that you can avail. So if you go on these websites, you will find a lot of information about the kind of businesses you can do. You'll get an idea about the kind of businesses that you can do and the technologies that have been developed and are available for commercialization. Now, when you have a microbe in hand, which you feel can be commercialized, what you want to do next is make a formulation. So earlier speakers also told you about what formulation is and how important it is. It is a very big, actually, it is a very big research area, formulation itself, because you have to make sure that you cannot apply the same carrier or the same formulation to all the bacteria. You have to make sure that your bacteria or your microbe, whichever you're using, your fungus or your bacteria, is compatible with that carrier that you're using. Now, what compatibility means is whether when you mix that carrier with your microbe, whether you are whether the microbe holds the capacity to grow inside the soil, also whether it retains the capacity to survive inside the soil, whether it retains the capacity to grow inside the soil. And even for the formulation, the shelf life of the formulation is also very important, because when the farmers buy it, they are not going to use the whole thing at a time, because these packets are available in, say, 1 liter per acre. What if you don't have 1 acre, you just want to use it in your small kitchen garden or something? So shelf life is also very important while making the formulation. So you have to do shelf life studies for the formulation. You have to look at the compatibility of carrier with your microbe, and whether after formulation the microbe is able to colonize the root rhizosphere or not. You have to look at that also. So these are some very important points that you have to keep in mind while making the formulation. Various carrier based, we categorize them into carrier based and liquid formulations. So when you say carrier based formulations, these are mostly granular carriers or wettable powders. Wettable powders, application wise also you have to see, like wettable powders are mostly used for seed coating. So when you coat your seed with certain biopesticides and then put in soil, it is considered that it will protect the seed from the pathogens that are present in the soil. So so you have to look at that, what kind of carrier you're using. So there are water wettable powders, there are granules on which you coat the bacteria. These granules are mostly used for field application. You just put the granules in soil. Now, while making the formulation, you also have to make sure that it is easy to use for the farmer. The farmer doesn't want to go through a lot of steps, to just apply biofertilizers in his farm. So that also you have to keep in mind. Liquid formulations these days are very, very popular, because you just put them in the water and put it in the drip and irrigate your farm with that. It is very easy to use. Liquid formulations, they also have higher shelf life compared to because they're not exposed like granules, right, or peat and all. So what happens in carrier based formulations, the microbes are exposed to environmental conditions. They are exposed to heat, they are exposed to UV. So they are more prone to not surviving. While in liquid formulations, this kind of problem can be solved. So liquid formulations are more preferable. Also these days, there are many more different kinds of formulations that are coming. Capsules, tablets, these capsules, tablets, gels, small pouches, what they do is they concentrate the quantity of bacteria or microbe, okay, microbial spores. So these are in concentrated form, so they positively affect your logistics also. these are easy to carry, these are easy to transport, and since they are encapsulated, micro-encapsulated, you have alginate beads, etc. Various polysaccharides can be used for micro-encapsulation. And if you are providing in capsules, they are protected against desiccation, etc. So these advanced formulations are also coming into market these days, and that is why the market of biofertilizer is expanding at a very good rate. Now, after you have a formulation in hand, you have to make sure that it complies to all the FCO or CI, whichever regulatory body it belongs to. You have to make sure that your formulation complies to the regulations of those regulatory bodies. So you have to have a quality control, a decent quality control laboratory, which has, of course, a laminar flow hood and microscope. These are two essentials of any microbial lab. Once you have once you have taken care of the quality control aspect of it, then you have to put your formulation to test. And putting it to test means putting it out in the farm. So it should be applicable in greenhouses, it should be applicable to kitchen gardens, it should be applicable in farms, and field trials are very important. As Dr. Sharma told you, that field trials can be carried out with the help of the agricultural universities or ICR institutes. They help you carry out these field trials, and you have to, it is mandatory also. So you have to send your products for field trials, and then you can come up with a good product. So these are some field trials of our products that we have carried out. Our farmers have now become so smart. And actually, as Dr. Sharma said, farmers are very, very smart. You know, they are actual scientists. They kind of divide their farms into treated and untreated areas, and they are able to register those results very well, very intelligently. They also report those results to us, and these are our designated farmers who we provide our products for field trials, and they give us very good results in tabulated forms and everything. So we are very happy with our farmers, and we want our farmers to be happy with our products. So we make sure that our products are high quality. And then we come to the packaging, labeling, and certification, etc. So packaging has to be something that is, you know, appealing to the farmers, because you the kind of customers that you are catering to, you have to look at the kind of customers that you are catering to. Very subtle labeling, subtle labels and subtle packaging is not going to be very appealing to the farmer community, right? They will look for packets that are attractive and that show what exactly the product is doing. So that kind of packaging is to be done. And labels are, of course, the labels are fixed because they have been prescribed by the regulatory body itself. We have to make the labels of our biofertilizers according to FCO norms, and the labels of our biopesticides have to be according to CIB norms. So these norms have to be followed. So you can't do much of you know, creativity in labels. Certain content has to be there. Then you have certifications. So on top you have this FCO certification by Ministry of Chemicals and Fertilizers. You have CIB certification, EcoCert certification, FCO and CIB. These are mandatory. These are the basic certifications that you will require if you want to start biofertilizer or biopesticide industry. So these two certifications are very, very important. Then there are EcoCert. EcoCert is something, it is for organic certification of your product. So organic certification, what that does is, EcoCert is also recognized outside India. So we have seen that for export of our products, EcoCert certificate is helpful. Then there is ISO. ISO is a certification of your facility, whether your facility is abiding by all the standards that have been set up for this kind of industry. So industry certification. And then there is BIS. BIS is a certification for your products, whether the products are complying with all the norms that have been set up by the government. OHSAS is a certification of good practices, whether you are taking good care of your employees or no. Their health and safety is also important. After all, this is a biopesticide and biofertilizer industry. You are using a lot of particulates in that industry for production. So you have to take care of your employees as well. So this is something very important, OHSAS certification. And DSIR certification is given to the industries, to the R&D research and development facility of the industries, and whether your R&D facility complies completely with the government norms or not. Then you are provided a DSIR certificate if they are satisfied. For marketing, you can go for dealer distributor network, you can go for direct retailing, you can sell your products on e-commerce websites these days. Then there are marketing partners. You can just provide technical and the other person will sell it under their own brand. So that is also possible. Marketing partners can also be availed. Now, this is something that I would like to touch upon. Patenting. Patenting in biofertilizers, whether you can patent a biofertilizer. In India, you cannot patent any plant, animal, or microbe which is available in nature in its native form, right? You cannot do that. So biofertilizer per se, the microbe itself is not patentable. In India, genetically modified microorganisms are patentable. And you see, it would be so convenient to come up with a, come up with a microbe that is very efficiently able to solubilize phosphorus. You can do that. You can just tweak around with the metabolic pathway of phosphate solubilization or a nitrogen fixation. You can just tweak around with that, or you can come up with the potassium solubilization pathway. So you can tweak around with the organic acid production pathway easily. But genetically modified or microorganism cannot be registered as organic input in India. So that is out of question in India. Okay, because recently I've read a report that Pivot Bio, which is a startup in America, has come up with a microbe that has genetically modified nitrogen fixation system, which is able to fix nitrogen even more efficiently, much more efficiently compared to the conventional products that are available in market. So in India, that cannot be done, and rightly so. You cannot just go on putting your genetically modified microbes in the nature. Who will control their population? I mean, biodiversity conservation is one of the most important focus of using biofertilizers, right? So if you're going to put genetically modified microorganisms in nature, in soil, their population you cannot control, right? So genetically modified microorganisms are not registered as organic inputs. However, you do have many options here to patent, even in biofertilizers. If you come up with some unique formulation, which is way more, which, kind of complies to these patentability conditions, subject matter, industrial application, novelty, inventive step. If your formulation, you feel that it is compatible, if it is complying to any of these, you can patent that formulation. So, or if you come up with a consortium which is showing synergistic activity, if you have that kind of consortium, and you make a formulation which has good shelf life and everything, then you can patent it. Then of course there's another way. If you come up with genetically modified microorganisms that are producing metabolites which will help in plant growth, say biostimulants, etc., then um, you can patent that microorganism, and you can register the, or get license for the product that is being formed by that particular microorganism. So these are the options that are available for you in biofertilizer market for patenting if you need them. So with that, I would like to end my presentation and thank you on behalf of my whole team for giving us this opportunity. And that's it. I'm done. Thank you.
Can you hear me, sir? Ma'am?
Yes,
Am I audible?
Yes, you are.
Thank you so much, madam, for your informative and very wonderful session. And I'm happy that you have included the patent aspect because I think it is one of the most burning topic which everyone speaks about when they are talking about formulations or bringing a product from lab to commercialization. So thank you very much for that. And I would like to ask another question, like, can the use of biofertilizers surpass the use of chemical fertilizers in terms of agroeconomy in present times?
In present times?
Yeah.
Hard to say. Hard to say. But in terms of economy, yes, because of all the new schemes that have been introduced by government, yes, I would say. Definitely. And especially the mandates. If you read in that slide, I have put that it is mandatory to use biopesticides along with pesticides. So yes, economically sustainable as well already. It is mandatory. Yeah, government has made certain policies which make the use of biofertilizers and biopesticides mandatory.
Well, I think the more we are aware about this and more awareness there is, I think we can definitely stop the use of chemical fertilizers.
We should. It is the need of the hour.
Okay. Thank you, ma'am. Do we have more questions? Dipali Shukla, ma'am, you want to ask some question?
I have a question, ma'am. Is there any application of biofertilizer in hydroponics?
Hydroponics, yes. Biofertilizers. We have done those experiments, but we do not have compiled results of it. But people are using. I have read certain articles where people have tried biofertilizers in hydroponics as well.
Okay. Thank you.
One question. Hello.
Yeah.
One question. When we use a biofertilizer or biopesticide in directly live condition in the soil, is that possibility they alter the soil microflora?
Yes, they do.
Maybe it may alter the bed side also.
Bad side? Why would they? It's highly unlikely. Why would? Because you are using the bacteria or microbes that are beneficial for the soil quality.
मैम सॉइल क्वालिटी के लिए बेनिफिशियल है, पर जो पहले से माइक्रो ऑर्गेनिज्म है जैसे कि माइक्रोराइजा या डिफरेंट टाइप के और माइक्रो ऑर्गेनिज्म जो सॉइल में है,
कंपैटिबिलिटी, हाँ, आप पहले कंपैटिबिलिटी टेस्ट करके फिर ही उसको इनपुट की तरह यूज कीजिए. That is also very important.
या मतलब जैसे कि पहले पेस्टिसाइड आए थे तो फ्यूचरिस्टिक प्रॉब्लम इसके बारे में अभी कहीं नोट की गई है या ऐसा नहीं किया गया है अभी तक?
बहुत सारे स्टडीज है regarding that. There are a lot of studies regarding that, and Dr. Sharma would have been able to answer that question very well, because I'm sure he has also done several studies, similar studies.
So only
And the purpose,
we must study the availability of local flora which is present already in the field. Based on that, the biofertilizers will be recommended.
exactly.
What are the natural strains are there? Based on that, we will recommend which type of biofertilizer can be used in that particular crop and in that particular area.
Okay. Thank you, ma'am. Thank you, sir.
Thank you so much, ma'am. I think there are no more questions, and if there are some questions, we will mail it to you all so that we can benefit our participants. So thank you very much, Dr. Ratnaparkhe madam, Sai Prasad sir, Dr. Sharma sir, for your beneficial presence and inputs. I hope our today's seminar has been very informative to not only academics but also prospective researchers and students who have joined us from all across the nation. I duly anticipate that our purpose of the seminar has been productive and fruitful. My heartfelt gratitude and thank you to all the e-learning department for their huge cooperation to make this a great event. May I now invite our head, Mrs. Pragya Goyal ma'am, to give her concluding remarks on this national seminar and propose the vote of thanks.
Thank you, Razia ma'am. Good afternoon to all of you. As all good things come to an end, so did this seminar on recent researches on biofertilizers from lab to commercialization. This seminar included prominent speakers from reputed research and commercial institutes of India. Dr. Sushil Sharma and Dr. S.V. Sai Prasad presented their keynote address on various research aspects of biofertilizers, while Dr. Supriya Ratnaparkhe highlighted the commercial aspects of biofertilizers as well as its patenting. Their talks enlightened us, and we could feel the passion with which they connected with us and with the subject. They have also motivated the students and our future researchers for research in this field, and they can even go for entrepreneurship from the ideas they have got from the speakers. We had 200 plus participants from all across the nation, that is from Chhattisgarh, Pondicherry, Hyderabad, Uttar Pradesh, Madhya Pradesh, Goa, Maharashtra, Guwahati, Jharkhand, just to name a few. We also had international participants from United Kingdom. At the end, I hope that you all feel enriched and have benefited from the seminar. I now propose the vote of thanks. On behalf of Department of Biotechnology, I take this opportunity to propose vote of thanks to those who have directly and indirectly contributed to the seminar. At the outset, I thank our keynote speakers, Dr. Sushil Kumar Sharma, Dr. S.V. Sai Prasad and Dr. Supriya Ratnaparkhe for accepting the invitation and sparing time from your busiest schedule to grace the occasions. Your ideas have enlightened our minds and have shown us a new path. This is surely going to encourage us in our future endeavors. We are thankful to Honorable Architect Achal Chaudhary sir, President IPS Academy, for his constant support and motivation. I would also like to extend my gratitude towards our Principal Ma'am, Dr. Premlata Gupta, for encouraging us to conduct a seminar like this today. I am again thankful to you, ma'am, for your kind support, suggestions and guidance that you have given to us. I also express my gratitude to Dr. Sanjay Nagar, Registrar IPS Academy, for his extensive cooperation and guidance. I am also thankful to the management of IPS Academy, without whom it would not be possible to carry out this seminar at national level. I also want to thank Dr. Sandeep Gupta and his entire team of AV Studio who worked behind the scenes to execute this event smoothly on a larger platform. We are indebted to them for their forever cooperation. This type of event cannot happen overnight, the wheels started turning many days ago with in-depth planning and bird's eye for details. For this, a special thanks to Dr. Rajesh Khande, coordinator of this seminar, and to a very active and dedicated organizing committee, Dr. Sultana Razia, Professor Preeti Baladatia, Professor Bhavita Kohli, and Dr. Deepali Shukla for their hard work for the past few days, unflinching support and coordination. I am also very thankful to all the heads of various departments for their remarkable presence and constant motivation. I would indeed be failing in my duty if I do not express our gratitude to all the non-teaching staff for their wholehearted support to make this program a successful one. A heartfelt thanks to our students for their active participation in this seminar and as volunteers. I would also like to thank all the participants for making the time to be with us today and hearing every speaker with full concentration and helping us make this event a grand success. With this, we move to the end of today's seminar. Thank you.
Thank you, Madam, for your wonderful vote of thanks. Once again, all our speakers, participants, faculties, staff of e-learning studio and our team on behalf of the organizing committee for your gracious presence on the national seminar on recent researches in biofertilizers from lab to commercialization. Your very presence is an enhancement of joy. Participants, kindly fill the feedback link which will be shared in your WhatsApp group. You will shortly get your certificates. I hope you all enjoyed the program as much as we enjoyed organizing it. Till then, goodbye. Thank you.